Hi, I'm Ricardo from the LM Institute, and I work in microbiome. So it is recognized as it's quite important for human health. Indeed, they provide the biochemical functions that, to degrade the food. And it's also involved in disease as a cause. So you have like Helicobacter pylori ulcers, or you also can affect the microbiome by another disease in the colon, like colorectal cancer or inflammatory bowel disease. So the goal of the field is mostly to predict disease state from the microbiome. And for that, people normally use the microbiome abundance, but it's quite difficult to use this data because there are like many differences even between healthy people. If you have different lifestyle, different diet, you have a different microbiome that's reflecting the data. So we wanted to add more modalities to see if we join them, we can get better prediction. So we joined microbiome abundance and taxonomy. Our results show that in the case of the variational autoencoders, if you take both of them in just a single study, it helps. It gets to squeeze more of the biology, and you get better prediction, and you get better representation in the latest space. In the opposite case, if you have multiple studies and you train your model in several of them, including from different diseases, you get a loss in the performance. So basically, the taxonomy is interfering, is causing like a loss on the domain invariance in the model, and basically we provide recommendations of how to separate the latest space to make possible to train these models in all the data, and to be able to use taxonomy to increase your performance. So if you want to see my poster in the first floor, we'll be happy to talk about it.
Thank you, Ricardo. Yeah.